Excellent. Well, thank you, and welcome everyone. Let's just get started. I'm going to be giving you really quite an overview of a number of. And tomorrow we're going to have Dr. Aaron Rose and Dr. Jim Keck diving into some of these models and looking into some of the more specific details of how these things can be used in your drug development pipeline. Now, Greg just gave us a really wonderful overview of how the Humanized FcRn platform can be used for your lead selection. And so what I'm going to focus on is the next stage is how we can take a look at these molecules in terms of efficacy and safety. Safety is in terms of related to cytokine release system, release syndrome. And really, we can use these models in a very sophisticated way to begin to ask and answer questions about looking at the balance between efficacy and safety and finding those molecules that have the best performance overall, hopefully accelerating your ability to have success when you get to the clinical stage of drug development. What I'll do first is begin with what exactly are we talking about when we say a humanized mouse, because this terminology is being used pretty broadly in the literature. And this is the reason why, because you have multiple ways that you can humanize a mouse. The first of which is genetic manipulation, as Greg pointed out for his models, where you're expressing ligands, receptors, growth factors, et cetera. And in some cases, these are the targets for your molecule, or they're supporting how these molecules perform in the mouse, as Greg demonstrated. But also there's growth factors that are necessary for the support of these immunological or immune cells that are engrafted into these mice. So even with that, even without any genetic humanization, there's two different ways that you can immunologically humanize a mouse. And one is with human peripheral blood mononuclear cells. And there's a wide range of approaches to setting up those mice. With irradiation, without irradiation, cell dosage has a factor. Different donor PBMC populations have different engraftment characteristics, and all of these things need to be characterized deeply. So it's not as simple as irradiating a mice and just injecting some cells. There's a lot of sophisticated background and preliminary information that you need to collect, in which the Jackson Laboratory has already accomplished and has a number of PBMC samples that are highly characterized, and we understand the engraftment characteristics of those cell populations. And then, of course, there's the cord blood-derived CD34 positive hematopoietic stem cells. This model requires ablation to get good levels of engraftment, and you get multi-lineage reconstitution. And so we'll go through some data really understanding the differences in the hematopoietic reconstitution between these two different models. And this is very important to consider because in addition to plain genetic humanization, PBMC humanization, hematopoietic stem cell humanization, we also have mice that are genetically engineered to express in human genes that better support hematopoietic reconstitution and engraftment. So they would be considered almost double humanized in that instance. So let's go through some of these different model platforms so that you can understand them a little bit better. Greg gave a wonderful presentation, so if there are those of you who have just joined us, I'm just going to present one slide talking a little bit about this genetically humanized FcRn mouse and really just talk about the highlights of it. And that is the fact that different therapeutically used, clinically used molecules like belatacept, ipilimumab, pembrolizumab, they have unique half-life characteristics. And if we look at the human half-life characteristics of those three molecules, as shown here, they do have different half-lives, okay? And if you put them into a regular C57 Black 6 mouse, you begin to see this breakdown of the half-life characteristics. And as Greg very nicely pointed out, in some instances you may see good modeling, but in other instances you do not. So a regular Black 6 mouse is not going to be reliable for modeling these PK characteristics. And of course, if you knock out the mouse FcRn molecule, then you have really quick clearance of those molecules. And you really need the presence of the human FcRn. So if you go back to the TG32 mouse, which is a knockout of the mouse receptor and transgenic expression of the human receptor, you begin to return to these characteristics of how those molecules perform in that mouse. And if you do a correlation analysis between the human and the TG32, you begin to see this really nice R coefficient, really showing the correlation between how that model develops PK versus the human. And again, if you do that comparing to the wild type Black 6 mouse, you have a lower value and you really don't reproduce those characteristics well at all. So let's go now and talk about immunological humanization and begin with talking about the platforms and going through the different platforms and building up in a way that you can understand the differences in how the different models are being used. So we start with the non-obese diabetic genetic background. This is the in-bed strain of which all these genetic manipulations are built upon. And one thing that's important to note about this is if you look at the innate immune function of this mouse compared to, say, a Black 6 or a BALB/C, they're deficient in complement. They have dendritic cells, macrophages, and natural killer cells, but if you put them in an assay and measure their function side by side to some of these other more common inbred strains, they just don't function at the same level as those cells. So there's like an intrinsic difference in immunological function of this mouse. And then if you put onto that mouse, you add in the SCID mutation, which is on the repair side of VDJ rearrangement. You have a lack of the ability to rearrange B and T cell receptors, so you don't have functional B and T cells. Then if you layer further into that genetically developed mutation in the IL-2 receptor common gamma chain, which is a subunit of the receptors for the cytokine signaling molecules IL-2, 4, 7, 9, 15, and 21. You no longer have functional receptors, and this is particularly important when you think about IL-15, and that will knock down the mouse natural killer cell development, which was a barrier to really good multilineage hematopoietic stem cell reconstitution in this mouse. So you have this highly immunodeficient platform receptive to the engraftment of these different human cell populations. Now, if we first begin to focus on PBMC engraftment, there's a really nice paper by King et al., which included Dr. Michael Brem and Lenny Schultz. And they were looking at PBMC engraftment, and they looked at a number of really important things like with and without irradiation, different cell doses, different drug treatment profiles, things like that. But also very importantly, Lenny created two unique NSG mice. One was completely knocked out in mouse MHC class 1, and a second knocked out in MHC class 2. And they definitively showed that it's the mouse MHC molecules that are being recognized by the human PBMCs and causing an accelerated xenogeneic graft versus host disease in this model. So using that information, they said, well, wouldn't it be wonderful if we had an immunodeficient mouse that we could put human PBMCs into, that we wouldn't develop graft versus host disease, and we could now start beginning to do more long-term studies. We could add tumors on board, and we don't have the complications of graft versus host disease. So what he did is he moved on and created a MHC class 1, class 2 double knockout. And this is the long extended nomenclature because he knocked out the NSG KB GB alleles to knock out MHC class 1, which was the alpha 1 and alpha 2 subunits. And then he knocked out NSG-IA for class 2. So it really has multiple genetic manipulations to knock it out, but it's a phenotypic knockout for class 1 and class 2, and that's why it's a phenotypic double knockout of class 1 and class 2. So what does the engraftment profiles look like, and what's the survivability, body weight, and all those different things if we do a side-by-side comparison of these two different models? So we irradiate these mice, so this is under irradiated conditions, and we look very early. We look very early at day 5. We can see that there's about a 6% engraftment, and we look within these human CD45 positive cells in the peripheral blood of these mice, we can then break it down further and ask questions about what are the different subsets that are engrafted at this early stage. And it's primarily dominated by human T cells as a measure of looking at CD3. So this is both CD4s and CD8s in here that are making up the bulk of this population. There are some CD19 B cells, some CD14 positive monocytes, some CD56, natural killer cells, and some CD33 positive. That's a more broad myeloid marker. Now, it's important to point out if you do a time course and you look at day 10, day 20, things like that, you're going to be seeing an expansion of the T cells and you're seeing a decline of all these other cell populations. So this is predominantly a T cell mediated engraftment model. So now if we look at a comparison between NSG and the double knockout in terms of body weight, we see that there's a rapid decline in body weight, indicating the accelerated development of this xenogenetic graft versus host disease in the NSG mice. If we compare that to the class 1, class 2 double knockout, we see that those mice are maintaining body weight and they have more long-term survival. So this drop in body weight is associated with this rapid decline in health of the mice. And of course, they reach a certain criteria and they actually need to be euthanized for animal welfare reasons. But it's important to note that we're not eliminating completely graft versus host disease in the double knockout, because there are occasional a mouse that will drop in weight and need to be euthanized. And taking a close look at the different PBMC donors and the amount of cells that are dosed, you can increase cell dosage and begin to see the development of graft versus host disease even in the class 1, class 2 mouse. But we have titrated and carefully characterized these PBMC donors so we know what levels that we can inject into these mice and understand how to use these mice critically in your experiments. So now let's go on quickly and talk about some of the more sophisticated genetically humanized models and how that influences hematopoietic stem cell engraftment. So in the regular NSG mouse, you get nice multi-lineage engraftment after you get those stem cells engrafted. But if you look closely at NK cells and myeloid cells, they're present there, but they're at very low frequency. And that's because these cell populations really require these human-specific growth factors to develop. So in a mouse model that we're calling the SGM3, they're transgenically expressing human IL-3, GM-CSF, and stem cell factor. And this improves the myeloid development pathway, but it also improves CD4 positive development and regulatory T cell development. But it still is somewhat lacking in terms of the natural killer cell population, which are an important immune target for immunotherapy. And so we also have developed this NSG IL-15 mouse, which is a human back expressing IL-15 at physiological concentrations and actually develops natural killer cells at a frequency in the peripheral blood of these mouse that very much mimics the human population. So if you do side-by-side studies where you're injecting the same stem cell donor in either NSG or the SGM3, and then you look 12 weeks later, which is when engraftment stabilizes in these mice, and you compare these different cell populations, you could see that overall, with those human growth factors, you improve overall engraftment at this time point, CD4 cells, regulatory T cells, and of course the myeloid population. And then if you do the same kind of study looking at NSG versus the IL-15, you could see that these other populations are very similar between these, and it's really the inclusion of IL-15 that improves the development of those CD56, CD16 positive human natural killer cells in those mice. Now, we've got this nice chart that you can take a look at later when you receive these slides. And the main point of this graphic is to show you that there are differences in these platforms in terms of the cell populations that develop. And this is used as a tool to help you decide which platform is most appropriate for answering your scientific questions. Now, the CD34 engraftment is going to be answering more sophisticated questions, whereas the PBMC engrafted mice is really going to be focused on the T cell populations and whether you're upregulating or downregulating the T cell populations. And whether you're looking at GvHD as a readout or you're looking at some other fundamental readout like cytokine release or tumor efficacy or something like that. So the PBMC platform, if you're really focused on T cells, is a really wonderful and very highly modular platform. We can set up this assay in a number of different ways. We can do it with single donors, single PBMC donors, so that you can compare your leads side by side. Once you got a more characterized selected lead, then we can move on to studies that use multiple donors in the assay. And you can begin looking at preclinical diversity of response and really giving you an idea of what you might predict to see in those phase 1 clinical trials. So we can talk to you about what your scientific goals are, and we can choose the proper host. If you want to use the mice and you want to develop GvHD, so you can look for immunosuppression by your molecule, we can do that. Or we can turn that on its head and you can say, okay, we can generate graft versus host disease. And what we want to do is maybe we want to activate those T cells. So you can actually look for acceleration of graft versus host disease to indicate that your molecule is activating those T cell responses. But then we could also move to the double knockout platform, and we can start looking at in vivo cytokine release assay. So we can look at your molecule in the absence of graft versus host disease, and is it going to activate or induce cytokine release? Or we can use this platform to say, all right, we've got molecules that are known controls that routinely develop cytokine release, and use that as a tool for anti-inflammatory discovery. Can you use it to suppress cytokine release syndrome in this molecule? So depending on what you want to do, there's a number of different ways that we can utilize this platform. And so let me show you a quick example here of looking at graft versus host disease models. So this is the NSG mice. They're irradiated. They're given 10 million human PBMCs. And then we're looking at the ability of abatacept or belatacept to suppress the development of GvHD. Just as a reminder, these are fusion molecules, CTLA fusion molecules that block costimulatory interaction between the T cell and the antigen presenting cell. In this particular study, they were used at 2 different dosages. These molecules do have different binding characteristics, but I do want to point out the differences in doses here as well. And I'm just going to give you a snapshot at the later stage of engraftment. You can use this model to look at day 5, day 10, day 20, and get a snapshot of the T cell activation status. Because what will happen when you put these PBMCs in is you have a loss of CD4 naive T cells. You have the accelerated development of CD4 central memory and effector memory cells. Then the CD8 T cell population becomes activated and the CD8 positive T cells expand, loss of those naive CD8 T cells, and an expansion of the T effector cell population. And that's very, very, very repeatable. And so what you can do is look at your drug treatments and look for things like with an abatacept at the 150 microgram dose given beginning prophylactically at day -1 and every other day thereafter. You can see that there's a retention of naive T cells, a reduction in CD4 T effectory memory cells. There's a better reduction of the CD8 T cells that are expanding out into the periphery. And at this later stage, you have a reduction at that higher dose of abatacept of the CD8 T effector cell population. So you can really look at these different T cell populations to really understand how your molecule is working in this system. Now, if we then think about this in the context of a cytokine release assay, we want to go through and develop the reason for why our group developed what we call this in vivo cytokine release assay. If we go back and we think about the more traditional assays that are being used to look at cytokine release today, as we evaluate these molecules, we've got the in vitro assays and we've got the non-human primate assays. And those are really kind of the gold standards. However, if we use the T-Genero 1412 example from 2006, this molecule was tested in both of those platforms, and neither of those platforms uncovered the cytokine release toxicity that was observed when this molecule was used in patients for the first time. So there is some unreliability there. So our development group, led by Jim Keck and his team at the Jackson Library, says there's got to be a way that we can do this better. So he went back to the lab and started looking at how to do this in an in vivo way. And the reason that in vivo is critically important is because it's not just the immune cells that are being activated in isolation. If we look at all the publications that have come out on this syndrome since this event, we know now that epithelial cells lining the vascular structure play an important role. So this is a cascading response where other cells and tissues in the bodies also produce growth factors that lead to this cascading response. So the in vivo response is critically important for measuring this type of activity. So what we're showing here is 3 different PBMC donors injected into irradiated NSG recipients. At day 6, we're dosing with an anti-CD28 conventional costimulatory monoclonal antibody. And then he developed its TGN1412 biosimilar, which, as a reminder, is a superantigen quasi-autonomous T-cell activator, which doesn't necessarily require T-cell receptor activation to lead to co-stimulation and activation of these cells. And what you can see here is a clear human cytokine release of interferon gamma, TNF-alpha, and IL-10. And there are donor-to-donor specific differences in responses, which is also observed clinically. And again, tomorrow, if Jim Keck is going to go into much more detail about these different responses, and I'm just going to highlight some of the data here. But what's also important about this in vivo model is you can develop a scoring system to look at clinical scores and body weight to get more sophisticated information about what's going on in the system. So the blue arrows indicate when the dosing was done, and we can look at within hours after that, you have the cytokine release, and you could begin scoring these animals for looking at an overall health of the model and develop basically a clinical condition of these animals. And you can see that with the higher cytokine release, it's shown in the prior slide in blue, these mice develop worsening accumulative clinical scores and a more rapid drop-off in weight, really demonstrating this systemic response that these animals are showing. Now we can take this a step further and we can begin doing things like putting tumors on board and really beginning to look at a balance between development of cytokine release syndrome and balancing that with tumor efficacy. And again, I'll give a brief example here, but Jim Keck is going to take that to the next level if you join us tomorrow, and really looking at dose ranging studies and things like that, and looking at the balance of looking at lead molecules and looking at the balance of these different responses to better have a more sophisticated way of looking at lead selection. And so this is modular too, because whether you're looking at a disseminated tumor model or you're looking at a solid tumor model, we can change up the engraftment profiles, whether it's PBMCs on first and then the tumor going on board or vice versa. We can look at monoclonal antibody therapies, we can look at bispecifics, we can look at a number of different things. So in this specific example, we're looking at a bispecific monoclonal antibody. It's an EGFR CD3 bispecific. So it's engaging CD3 T cells and EGFR on the tumor. And this is a triple negative breast cancer cell line that expresses EGFR. It's labeled with luciferase so we can track this model very quickly in the peripheral of the mouse model using a luciferase detection assay, a luminometer. And what's really key and important here is we've got control molecules, we've got the bispecific, and then we included Keytruda, which is a PD-1 checkpoint inhibitor, to see if we could enhance that T cell function once that bispecific activates the T cell population in the context of responding to the tumor. What's interesting here is there was a control set of animals where there was no tumor injected, and there was no cytokine-mediated release. It was only in the presence of the bispecific alone or the bispecific plus Keytruda that we had strong interferon gamma and TNF-alpha release 6 hours post-treatment. So in the presence of tumor, you've got the interaction of the T cell, and you've got T cell killing, and you've got this cytokine release response, and you have clearance of the tumor. Now, what's also interesting here is with the anti-CD28, which was a control not expected to lead to tumor killing, we also think about the mechanism of anti-CD28, which can activate these T cells and can lead to a cytopenia or loss of T cells in the model. And this loss of T cells allowed an accelerated outgrowth of the tumor in the model. Now, we could also include clinical scores and body weight to tell us more about this, what's going on here. So it didn't look like the addition of the checkpoint inhibitor really led to an increase of cytokine release, and it didn't really add to tumor clearance. But we can see that if we look at the clinical scores, those mice were really not doing better. The accelerated activation lead to a decline in the health of the animals as indicated by accumulated clinical scores after the dosing. With the anti-CD28, we saw that these mice developed late developing clinical scores, which had nothing to do with the cytokine release, but had to do with tumor burden. So after dosing, we see a decline in body weight with the bispecific and the combination therapy, and we see this late development of body weight loss due to the tumor accumulation. So there's a lot of information that can be gained from this model if you really include clinical scores, body weight, cytokine release, tumor efficacy, and there's a lot of information that can be gained. Let's go on now and talk a little bit more about hematopoietic reconstitution with cord blood-derived hematopoietic stem cells and putting them into these different models. We've talked about the different cell populations. So the platform that you're going to choose for your efficacy studies and lead selection studies is going to be based on the population of cells that you're more interested in looking at in terms of your mechanism of action of your molecule. So obviously with PBMCs, you're really T cell focused, but there may be other targets like NK cells and myeloid populations, regulatory T cells. They all play a sophisticated role in tumor development or autoimmune disease or whatever it is that you may be studying. So we can advise you on these different platforms that might be most appropriate for your molecule of interest. And the other thing that I need to point out here, also in terms of moving into these efficacy studies, is the Jackson Laboratory is humanizing these different platforms on a monthly basis. So these engraftment models, you don't have to wait 3 months for them to be engrafted and validated. They're ready to go off the shelf and can be incorporated in your studies right away. So this is also very modular. There's a number of different tumors that can be used, but you can co-engraft tumors with these, and you can look at monoclonal antibody therapeutics, bispecific therapeutics, and also for those of you who might be also interested in chimeric antigen receptors, I'll give you some data, some small amount of data here looking at chimeric antigen receptors in terms of T cells, but we're advancing into CAR-NK, CAR-M, all these different things. And we can look at things about tumor efficacy, clinical scores, survival characteristics, and even cytokine release in certain instances with some of these models. So here I'm going to show you a data set focused on the humanized NSG with solid tumor engraftment of the MBA-MB231. This is a solid tumor that's a triple-negative breast cancer engrafted into the breast cancer tissue of these mice. And then we're looking at treatment of these mice with the EGFR CD3 bispecific again, but we're looking at a prophylactic treatment trial versus a therapeutic trial. And the primary difference here is the size of the tumors and when the beginning of the dosing occurred. So if you look at tumor size at about 100 millimeters cubed, and we look at the dosing schedule that was about 2 weeks of dosing, this is what we call a prophylactic trial where the tumor is relatively small You begin dosing early when the tumors are small, and you can look at clearance of the tumor. This is the average data, and this is the individual mice shown over here on the right. Versus a therapeutic trial where you have a larger bulk of tumor growth. The tumors are much larger, and you have to do a longer dosing strategy. But if you do, these bispecifics are very efficient of taking the T cells that develop in this multi-lineage reconstitution model and getting those T cells to respond against that tumor. If you are interested in natural killer cells, you can look at the CD34 humanized NSG IL-15 model and look for NK-mediated antibody-dependent cellular cytotoxicity. You can take the PBMC, the CD34 engrafted humanized mice. You can engraft them with an NK cell resistant Dowdy tumor cell, and then treat them with rituximab. Rituximab being an anti-CD20 monoclonal antibody used clinically to clear B-cell lymphomas from patients. It does have ADC killing as one of its mechanism of actions, so you can treat these mice with rituximab, and we have donor A, donor B, and donor C, just to really take a sophisticated look at donor-to-donor differences in responses, because this is a key thing not only for the PBMC model, but for the CD34 engrafted model. There are donor-to-donor differences in responses, so it's important to think about that as you look at the responses of your molecules. So in my last data set here to wrap up, I'm going to switch gears a little bit and talk about the NSG SGM3 model and look at CAR-T studies in this mouse. And I want to use this as an example because it's a really sophisticated study, and if you haven't seen this paper, I would encourage you to take a look, because I'm just going to touch on a few aspects of what they did, but they really did some really interesting and sophisticated work in this paper. Now, they chose the SGM3 because I said earlier they have more advanced T cell development. In the T cell development in all of these models, the T cells are going to be educated on the mouse thymus. So when you think about autologous CAR-T, they wanted to take these T cells out of HSC engraft-treated mice so that they could ex vivo transduce these T cells and put them back into CD34 engrafted mice, so that it was an autologous model in the context that the T cells are going back into these mice. They see the mice as self, and they don't have to worry about graft versus host disease. Now, they took these T cell populations and they developed two populations, one specific for CD19 to knock out B-cell lymphomas was their ultimate goal. And also they developed CAR-Ts that would recognize CD44 on myeloid cell populations, so myeloid-derived leukemias. And they first asked the question, well, can these CAR-T cells knock down the myeloid cell population and the B-cell population that naturally are produced in the CD34 engrafted mice? So if we looked at the CD19 positive cells or B cells treated with the CD19 CAR-T, we can see that they very rapidly knock down those B cells. And then if they looked at the myeloid population treated with the CD44 CAR-T cells, they very significantly knocked down the myeloid population, those cells. They did go on to put in leukemia cells and saw a clearance of leukemia cells as well. But what's also interesting about this model is they looked at cytokine release in this platform. And the black is the non-humanized mice. So you need the presence of those humanized target cells to develop the cytokine release syndrome. And they saw loss of body weight with both of these CAR-Ts. We saw the upregulation of human interleukin-6 as one of the key molecules that's released in CRS in the human clinical situation. The mice developed fevers, and they also saw liver damage is indicated by increased serum amyloid A. So it was a very sophisticated paper that also went on and looked at treating these mice that had developed a cytokine release syndrome with anakinra, which is an anti-IL-1, and they looked at the suppression of neurological toxicity in the model. So it's a very interesting and very sophisticated paper looking at how these this model could be used to look at a really broad spectrum of readouts for their drug treatment program. So with that, I'm going to wrap up and say that, remind everyone that there's a number of preclinical platforms that Jackson Laboratory has available for you. There's a number of different things that we can do with you to help you with your drug development program. And with that, I will just stop there and say thank you very much.